Inside your cells, proteins function as physical, nanoscale machines. They are the biological actuators that execute every movement and chemical reaction in the body. To understand how these machines work, scientists have traditionally followed a strict one-way pipeline. It begins with a linear sequence of building blocks called amino acids, the fundamental code of biology. That string of code folds into a highly specific 3D structure. The exact geometry of that shape is what determines the protein's ultimate biological function. This pipeline means science has largely been an exercise in reading. We can observe the sequences nature has already built, but we struggle to write our own instructions from scratch. Imagine we need a tool that doesn't exist in the natural world, a protein designed to seek out a specific breast cancer cell and deliver a drug directly to its surface. We can't simply guess the right amino acid sequence to build it. A single protein can have hundreds of building blocks, and the mathematical combinations are so vast that finding the right one by trial and error is a statistical impossibility. Researchers realized they needed a system that reverses the entire paradigm, starting with a desired function and reverse engineering the brand new sequence required to achieve it. Bridging this gap requires teaching generative AI to natively speak the language of biomolecules, allowing us to compute a path from a target goal back to a genetic sequence. Most of us know diffusion models as the technology behind AI-generated images. They work by taking clean data, corrupting it into random noise, and then learning to reverse that process to construct a clear image from scratch. But biological data isn't made of blendable pixels. Protein sequences are discrete units, individual amino acid letters that must be placed in a specific, non-continuous order, much like words in a sentence. This led to discrete diffusion. Researchers corrupt a sequence by iteratively masking out amino acid letters until the data is obscured. The AI is trained to reverse this. It looks at the blanks and step by step guesses the missing letters to make the protein functional. By repeating this process across millions of examples, the model moves past simple memorization. It begins to learn the fundamental rules that govern how biology is structured. While structure-based beta models exist, they are limited by a data bottleneck. We've only mapped about 300,000 3D protein structures, and those examples are heavily biased toward a few specific shapes that are easy to measure. Sequence data offers a much deeper well. Scientists have access to over 50 million sequences that span the entire evolutionary history of life on Earth. This diversity is the foundation for EvoDiff, a Microsoft research model trained on that massive evolutionary dataset to generate entirely original protein sequences. Researchers use a technique called biological prompting. You provide the AI with a small functional core, like a sequence binding to calcium, as a prompt. EvoDiff conditions on that prompt and computationally in-paints an entirely new, stable protein structure around that core. This indicates that generative AI is moving beyond just mapping the natural world. It is now actively drafting novel biological solutions that have never existed in nature. Designing a protein on a screen is a computational success, but it doesn't matter unless those digital designs can actually survive and function in a physical cell. To prove the concept, researchers took the sequences generated by EvoDiff and had them physically synthesized in a laboratory. The experiments confirmed the designs worked. These synthetic proteins were structurally stable and successfully performed their intended tasks, such as binding to targeted calcium ions. This bridge between computer science and biology proves that we can reliably use AI to design custom biomolecules for real-world applications. This creates a closed-loop system of discovery. The AI proposes a new protein, the lab tests it, and the experimental results are fed back into the model to make the next generation of designs even more accurate. However, a system capable of designing any biological function is a double-edged sword. The same engine used to create life-saving drugs could theoretically be repurposed to engineer harmful pathogens. Because of this dual-use potential, researchers and policy leaders are now prioritizing biosecurity regulations and ethical frameworks to ensure the technology is developed responsibly. If managed correctly, this approach will radically accelerate our ability to discover precision therapeutics and solve health challenges that were previously out of reach. To continue exploring the cutting edge of data, AI, and hands-on tech projects, be sure to check out the Learn by Doing with Steven YouTube channel and other platforms. And for deeper dives into the tech shaping our future, Tune in to the Stephen Data Talk podcast, available right now on YouTube Music, Spotify, and everywhere else you get your podcasts.